So a couple of announcements before we get started. How many of you are seniors and thinking about graduating? All of you signed up for graduation? Should be a fun party. It's going to be down at the CLSB. We're trying to have music, negotiating arm wrestling with OHSU about doing that. Free food, yes, free food. Seeing professors dressed up in funny clothes. Good opportunities for selfies. The opponent, but I won't be asking any questions. So should be a lot of fun. You can register on the biology department website, and you can get a free mug. Cool. And a t-shirt if you want one. So look at all these advantages. You know, no reason not to. It's on the 12th, so the last day of finals week, Friday the 12th, from I think 11 o'clock, if I remember correctly. So that's announcement number one. Announcement number two, having to do with all our friends, the clickers. Those people who were here last time saw that your clicker worked because we all, got to see how those were going. But unfortunately, my computer, this one, is not talking to D2L and clickers. So please, if you don't have scores listed on D2L for your clickers, just email me your Odin username and your clicker number, and I will manually input those. There are about 30 of you for whom that's the case. So please go ahead and do that. That should reduce the number considerably, and then I can upload all of your scores. At least that part seems to work. So please, again, email me the number on the back or inside if you've got an older one, and your Odin username. And I will connect those two so you will be appropriately registered and so you get all of your clicker points. Okay, questions about clickers and so on and so forth. So again, please send me that if you're not already there. For some reason, the first time I downloaded it, it worked, and ever since it's not. As we were talking about earlier, technology, it's such a wonderful thing. But of course, we still have our clicker questions. And the first one for today is, which of the following is the difference between dengue fever and hepatitis C? The vector, the number of open reading frames in the genome, the presence of an envelope in the virion, the requirement for host proteases, the requirement for viral proteases. Quick, Ben. Make your vote. 4, 3, 2, 1. So, how many open reading frames are there in both dengue and hepatitis C? 1. Envelope, both of them. Host proteases, which one is it? The furin protease, viral proteases. Yeah, all of these things need viral proteases. And the newest hep C drug targets the viral protease. So that leaves us with number 1. Yay, A. So that is correct. Second question for today. Which of the following causes the most deaths per year? Yellow fever, dengue fever, West Nile, hepatitis C, or Ebola? I should say human deaths per year. Human. 5. So in theory, clickers points are not supposed to be attendance points, but that's the way it seems to be. Yes, it is dengue by far and away. That's why so many people are interested in trying to deal with dengue fever. It would be really nice if we had the same kind of drugs for treating dengue as we do hepatitis C, but again, unfortunately it's an economic thing because most of the deaths from dengue are not in the developed world, they're in the developing world. Same is true of yellow fever. It's actually kind of hard to get a good number in terms of the deaths of yellow fever, but it's clearly considerably lower than dengue. There's also a pretty good vaccine out there, which probably is one of the reasons for that. Dengue fever vaccines, of course, are coming online. Hep C, there are really, really good drugs for, really, really expensive drugs. And Ebola, yeah, doesn't even blip relative to the number of deaths that we've had from dengue. Of course, it's not necessarily what you hear in the media, and what we'll be hearing about today in terms of the coronaviruses are also a very different kind of number, and again, media. So today, we're going to talk about coronaviruses. Adios, those of you who have to run away, and welcome those of you who just got here. But the coronaviruses are really fascinating from a molecular virology point of view, basically because they're way bigger than any self-respecting RNA virus should be. And this is a really interesting molecular question that basically only a handful of people cared about until about a little over 10 years ago. And of course, that's because of the emergence of SARS. Mostly, people studied coronaviruses because they thought they were causing most of the common colds. What does cause most common colds? Rhinoviruses, also positive strand RNA viruses. But no one again really would have cared much about SARS, or say coronaviruses, until SARS. And what's really interesting is there's a second, apparently independent outbreak of coronaviral disease, pretty nasty coronaviral disease, MERS, which has happened literally just in the last couple of years. So key concepts as far as the molecular virology of these coronaviruses are concerned. The receptors, really curiously, most of them, but not all of them, are proteases, which is really kind of bizarre. Why would you have a protease as a receptor? But lots of cells need to chew things up on the outside. So these are extracellular molecules, and it's a pretty good example of viruses are going to bind to whatever things they can find on the outside of cells. And again, lots of cells have these kind of proteases. There's a lot of discussion about whether proteolytic activity is actually important for virus entry. Because if you think about the structure of virions, there again, as I've mentioned probably far too many times now, they're in a metastable state. They've got to be stable enough outside the cell, but then be able to be degraded so that the genome can be released on the inside of the cell. And so potentially, and this is highly speculative at this point, using protease receptors is a way to do that. Frame shifting, this is not having to do with, again, thinking about shape shifting, whole zombies, et cetera, et cetera. This has to do with translational changes. And so a lot of what we have to do when we're talking about all kinds of RNA viruses is how you do translation. And that's going to be all a little bit different, and particularly important for these coronaviruses. And the main thing here is that there are a number of what are these called subgenomic RNAs, which are really important for making different parts of the RNA genome. Again, I mentioned these ginormous genomes. In a ginormous genome, you really want to be throwing away most of your peptides like you do with the picoRNA viruses and the flaviviruses, because translation is a really expensive thing to do. So that's in terms of the molecular aspects of coronaviruses. Now I'll talk a little bit about reservoir species, because this is really important for emerging diseases, and particularly things like SARS and MERS, which seem to pop up out of nowhere, certainly in terms of humans. So as usual, we'll follow our pretty standard outline here. Where do they come from? Which again is mostly having to do with disease. Talk a little bit about the structure. This was the most exciting thing about coronaviruses when people started to look at them, were these projections that they have here on the outside of the, a little hard to see on this particular projector, but all these jewels at the end of little stalks, as if you would have on a crown. And so that was what gave them their particular name. Binding and entry, again, we talked a little bit about that. Mostly it's a membrane fusion event, not too terribly different. The main different thing, and surprise, surprise, is in red, has to do with not so much genome replication, but really about the messenger RNA production, because that's very, very different in these still positive strand RNA viruses relative to anything else we've talked about so far. And then, how they get translated and get back out. So coronaviruses, at least until very recently, most people were interested in these because they caused colds. A number of animal viruses, which are relatively nasty, some canine and feline coronaviruses. But for the most part, people didn't really care that much until, of course, SARS coronavirus came along. And then everybody went nuts. So this, some of you probably remember this. It wasn't that long ago. That this was all over the news, everywhere, people wearing masks, et cetera, et cetera. So what was the disease? Again, mostly common cold and animal viruses. Almost everybody studied murine hepatitis virus, which, yes, did give mice hepatitis. Who cares about hepatitis in mice? All of us, yes, no. If you have a mouse colony, actually, it's a big deal. But other than that, nobody really cared. But it replicates in cell culture really nicely. And so that was the reason that people were using these murine hepatitis viruses. And then, of course, first reported in February of 2003, so almost exactly 12 years ago now. A month later, there were over 1,000 cases. And then this was originally severe acute respiratory syndrome, so it was a disease. And they discovered the virus in May. So February to May, how many months is that? 3 months between a brand new disease and actually discovering the virus. So that tells us where we've gotten to, even 10 years ago, oh my God, 12 years ago, in terms of discovering the etiology for disease. What's causing the disease? So it was a really amazing progress in, literally a couple of months between new disease and figuring out that it was a coronavirus, which was kind of a surprise because everyone thought, sort of minimal kinds of diseases which are happening there. So what was SARS? Again, severe acute respiratory syndrome. Main problem is that people basically couldn't get enough oxygen. And the death rate was about 10%. So people who came down with the disease, about 10% of them ended up dying. And this was in developed world. In fact, there were some big outbreaks in Canada in particular. China was the real epicenter of this. There were 8 cases and 0 deaths in the US, but that's probably just a statistical kind of thing here. How many SARS cases have we had here? Yeah. How many Ebola cases? Sorry. Yeah. Well, one real one that was actually here, but otherwise it's been 4, and only one of them has actually died. So how do you get SARS? Basically direct contact, as we were talking about before, aerosols. So anyone who's sitting next to anybody who doesn't have empty seats all around them would be exposed. So the real question is, where did it come from? Where was it before 2003? And so there was a lot of discussion about where it could have come from. The original outbreaks were in China, and where people in China could have gotten it from. At first, people thought they were civet cats, which are these animals used as pets, but also used as food. Then people thought that it could have been a bat. And this gets to the whole concept of a reservoir species. And we'll talk a lot more about this when we talk about Ebola and the filoviruses a little bit later on. But basically, this is where the virus hangs out. And when I like to think of this from much more of the viral perspective rather than the human perspective. Who cares about humans dying of these things? But clearly, humans are actually kind of a dead end as far as SARS is concerned. And as we'll see at the bottom here, the last updates on SARS were about 10 years ago. So clearly, humans are actually a really bad idea as far as SARS is concerned because it's basically died out. It's not there anymore. On the other hand, in these what people call reservoir species, this is where these viruses persist. And again, all these viruses are dependent on a host to be able to replicate. So they have to have somewhere where they're replicating. And in general, if you cause your host really nasty disease and in a worst case actually kill them off, then that's not very beneficial as far as passing that virus along. So generally, these reservoir species are some kind of animal, or of course you can have plant, whatever, host species, which then maintains the virus over long periods of time with no detrimental effects, and in some cases maybe, and again this is a pretty big maybe, some positive effects that are being maintained in them. And so the real place that you have a virus-host interaction is going to be in these so-called reservoir species. People care about the reservoir species people because that's usually where a lot of these diseases are coming from. And it's pretty clear that that's the case in many of them here. So we'll look more at the reservoir species in just a second. Just as a comparison here to have a bit of an idea in terms of how much coronavirus work was going on, in 2002 there were 130 publications in Medline, and 2003 there were 620 publications there. And 3,876 citations since 2003. I haven't done the Ebola math yet, but number of deaths, about 800 relative to number of papers that are published on it. So this is, I think, one of the highest papers to death ratios that I know of. So let's look a little bit more at the reservoir species specifically for SARS. Turns out that 50% of bats in the Philippines have coronaviruses that are associated with them. None of them seem to have any particular ill effects for having those coronaviruses, hosting those coronaviruses. Bats are really an amazing species, or say all of the bats are a really amazing group of organisms for all kinds of different reasons. A, that they can fly, of course, but there's some really interesting aspects of their immune system which may allow them to keep lots of viruses in them for long periods of time, and maybe even some of these viruses are, again, providing a positive effect in terms of how you can keep the virus-host relationship for long periods of time. So actually just a citation of that article here. Bats in the US also, almost all of them have, or about 50% or so, have some kind of coronavirus that's associated with them. Why do we care about bat coronaviruses? Well, basically because if you look at a phylogeny, and this is now, again, just looking at sequences. So sequences of course, we mostly care about this one down here, the human SARS virus. If you look at sequences of this human SARS virus, it's actually very similar to SARS virus associated, or I should say just coronavirus, associated with civet cats. And so that's why people at first thought that the transfer to humans was from the civet cats. But it turns out that not very many of them are in fact infected with these viruses. They probably actually got it from a bat originally. And part of the reasoning for that is, of course, there's a bat virus, which is quite similar to these guys. But if you look at all the rest of this phylogeny, you'll see there are bat coronaviruses, COVs, all over the place, and huge amounts of diversity. And so one of the things you can do with these kinds of phylogenies is basically build back. So here, common ancestor of the virus, which generated the SARS virus in human and SARS in bats would be, sorry, in civet cats would be here. Common ancestor now between bats and all of these guys is back here, etc. There are, however, a couple of problems with this particular phylogeny. Anybody notice any of them? Okay, one is a lack of outgroup, although they did have an outgroup in this particular paper. What else? think I talked about this before. Some of the numbers are extremely low here. So 35, and particularly here in this grouping, 37, 35. What that means is that when you compare all these sequences and you put them into their treeing programs that say this is how they should be related to each other, 100% of the time you get these three together. So clearly these three viruses are pretty closely related to each other, at least at a sequence level. 37% of the time it comes out together with this bat virus. But that means that 63% of the time it comes out together with something else. 35% of the time here. So yeah, it's grouping together here some of the time, but not always. So I think this particular phylogeny is not completely definitive that whatever viruses we have here really definitely do come from bat virus. Clearly closely related to these bat viruses, but there may be some other ones here. The other thing you need to bear in mind is that these lines here, the horizontal lines, represent sequence change. And the longer any of these lines are, the more sequence change you have. So you have to be very careful about things that have very long connecting lines relative to each other. But there certainly is much more diversity in these bat viruses than there is in any of these human viruses. And that means that it's quite likely that the ancestor was something similar to a lot of these bat viruses, and that there's clearly a lot more diversity in the bat viruses than there are in any of these SARS viruses.
So is it more likely that with a long line and a low number that there was some kind of recombination event between two viruses, or is it more likely that we just haven't sequenced any of the intermediates there?
Right. So the question is, what about the long lines, and how do you generate these long lines? Basically, sort of paraphrasing your question. And the answer to that is we don't really know. Certainly, all of the tree, well, at least this particular tree, was put together assuming no recombination, so just individual point mutations. And that's the way you make the alignment. You can make alignments in different ways, some of which will take into account recombination. It's a lot harder to make trees if you take into account recombination. And as we'll see, recombination is really rampant in these viruses. It's actually probably part of their replication process. They have to undergo recombination. So that's one aspect here. But it's distinctly possible that there was something else that we just haven't sampled as far as that's concerned. So yeah, open question. Okay, so what do these guys look like again? We've already talked about, we looked at one example right at the beginning. Crowns, they're clearly enveloped virions. They have this membrane that the crown proteins are sticking out of. Mentions here, and as you'll see later on in the textbook, a helical nucleocapsid. I can't see any helices in any of these EMs, but if you just purify the nucleocapsid away from the envelope, these are clearly helical nucleocapsids. And we'll see for the negative strand RNA viruses next week that a lot of these have helical nucleocapsids. And helices are great, again, if you think about them from the structural point of view. Single subunit, associating with all the other subunits in exactly the same way. What's the big problem with those helical symmetry? The ends. And the ends are then usually not protected. Well, if you're inside an envelope, that's not as much of a big deal. And so you can probably get away with having these helical nucleocapsids on the inside without. There seem to be some that might be icosahedral, and there's even, we'll look at the matrix protein or membrane protein in just a second. That seems to form a sort of icosahedral structure as well. It's a lot easier to look at this in a cartoon fashion. Now you can clearly see that it's helical. But again, that's because it's a cartoon rather than anything else. And then we have the spike protein on the outside. That's projecting on the outside, just like a bunch of proteins we've already talked about in terms of interacting with receptors and causing fusion. It's a trimer, and then there are conformational changes in that trimer that will give you membrane fusion between the viral membrane and the host membrane. in many but not all of these coronaviruses, there's also an HE protein. It's actually not shown in this cartoon. It's the hemagglutinin esterase. Hemagglutinin, what does that bind to? Sialic acid residues. These are the sugars on the outside. Esterase here chops off part of that sialic acid and is probably important for release of the virion once it's being budded, once it's being released from the outside of the cell. Very similar to HA and NA in the influenza viruses. The most abundant protein that you find in all of these virions is the M protein. This is, I would still call it a matrix protein. They call it a membrane protein. Again, different virologists from different communities working on different viruses will call their protein something else. But here it really seems to be serving as a connection between the spike protein and the nucleocapsid. That protein, the M protein, does have direct interactions with spike and direct interactions with N. N is the protein which is binding to the nucleic acid. So it really serves as sort of a bridge between the two. And in a few cases of coronaviruses, again, not SARS, not the murine hepatitis virus, but a few of these coronaviruses seem to have much more spherical structures. So unlike these blobs here, they do look more like this. And in the case of a sphere, not surprisingly, these have icosahedral symmetry because that's pretty close to the sphere. And so some of the M proteins and some of the viruses seem to be able to do this. A few of these have a small envelope protein that may be involved in budding. And we'll talk about some of the budding enzymes, or sorry, not enzymes, but proteins important for budding when we talk about HIV a little bit later on. What do these guys bind to? Very often peptidases. There are lots of different groups of coronaviruses. It's not important which ones these are. What is important that we're binding to lots of different proteases. And they're not all the same ones. Aminopeptidase, angiotensin converting enzyme. Again, all of these are proteases that the viruses are associated with and may be important for virus entry. Again, there are lots of arguments about whether that's happening. And it's not just these. There certainly are some examples of other ones, and not surprisingly, sialic acid seems to be important because you have this hemagglutinin esterase on the outside of the virions. How do you get fusion? Fusion happens basically very similar to fusion that we've talked about before for our flaviviruses last time, either in the receptor-mediated endocytosis, but in the case of the coronaviruses, this happens very often at the plasma membrane. And so this is one of the reasons why people think that binding to these protease receptors is probably really important for having proteolytic activity and then getting entry inside the cell, because it doesn't seem to be a real obvious pH-dependent change that happens. It's much more of a receptor interaction causing then a conformational change. But that conformational change looks basically identical to what we looked at for dengue and what we've looked at already for HIV in terms of pulling membranes together and then allowing the release of the genome. What is that genome? It's a positive strand RNA, and it's big. 30-odd thousand some, and it depends on which particular coronavirus you're looking at. Anywhere from, I think, the smallest are about 15,000 to the largest are, again, over 30,000. These things are enormous. And this was a real surprise when people first looked at them, because we haven't talked too much about this, but the RNA-dependent RNA polymerases have a big problem relative to DNA polymerases, and that is they make a bunch of mistakes. And it's usually about 1 in 1,000 or 1 in 10,000 nucleotides. Well, if every time you replicate your genome, you put in different nucleotides, that's going to be potentially causing lots of problems as far as genome replication is concerned. And anybody, again, I was asked this a couple times. There are only 1 or 2 of you, I think, that listened to TWIV. But a couple of weeks ago, they had a TWIV where a guy who was working on coronaviruses asked his virology class, how many of you would be willing to take a vaccine of a virus that had a much higher mutation rate than the normal virus? Why? Why am I interested in none of you? Because you don't trust me. Vaccines. Why? Well, but if you think about mutations, what are most mutations? Very deleterious. Very deleterious for virus replication. And so if you've gone up 10-fold or 20-fold in terms of number of mutations that you make per replication cycle, you end up losing a huge amount of virus replication. And one of the things he points out there is there's a lot of scientific evidence for that. Now, why did some of you not want to take this? You're worried about reversion. Right, so reversion to a wild type. Any examples of that you know of? So maybe in polio, but the frequency there is ridiculously low, and that's at the normal mutation rate of polio, as opposed to a much higher mutation rate. So yeah, I think it's an interesting thought experiment, but one of the reasons for that actually has to do with some of the experiments that they've actually done here. So if you look at this genome, again, it's huge and way bigger than anyone would expect to be able to be maintained throughout virus replication, because 1 in 1,000, 1 in 10,000 nucleotide changes, that's way too high to be able to maintain if you think about how much you need to replicate here. So we'll see why that is in just a second. This genome is really kind of interesting, and it's kind of flipped relative to the other positive strand RNA virus genomes that we've seen before. Here we have the non-structural proteins that are at the 5' end, and all the structural proteins which are down here at the 3' end. There are multiple open reading frames here. Okay, that's one clear difference. It's not just one like we have with the flavies and the picoRNA viruses. Each of these structural proteins down here at the 3' end are individual genes with individual start and stops. So they're different open reading frames as far as that's concerned. However, not unlike all of our picoRNA and flaviviruses, we've got one big polyprotein, and that's what's present here, gene 1, at the 5' end of the genome. Normal cap, normal tail. Again, positive strand RNA virus replicating in the cytoplasm. What does that tell you immediately about some of the proteins in the genome? What, do you have to have one activity in particular? I'm hearing muttering. So, where do you get caps from? Where are caps made in eukaryotic cells? In the nucleus, because that's where you have transcription taking place in eukaryotic cells. So, having a cap structure here means what? That's what I should have asked as a clicker question. So, you have to have a capping enzyme. One of the viral proteins has got to put that cap on, because the capping enzymes are all in the wrong place. So you have to have a capping enzyme. Yeah, sure, you've got to have an RNA-dependent RNA polymerase. That's, that kind of goes without saying. But just the presence of a cap here means that you have to be having a capping enzyme, which is there. Yeah, in the back? No. I'm sorry to keep beating on this whole capping thing, but when we talk about the negative strand RNA viruses, there's also some really interesting aspect of caps there as well. The poly A tail, that, can come from two different places. What would they be? It's in the genome. It's actually coded for in the genome, which is what we see in all the picoRNA viruses, or at least the polio virus. Some of them have the hairpin loop structures. What is another possibility? Could have its own polymerase. Could have its own poly A polymerase. That's definitely a possibility. Turns out not to be the case here. This is in fact encoded in the genome. But there are some poly A polymerases which you do find in viruses that we'll talk about right towards the end of the class. So cap and tail, big polyprotein. What do you have to do if you've got a polyprotein? Chop it up into little pieces. There's one other thing about this particular polyprotein. and that is, you can notice it's offset here, set up and down. It turns out it's one gene, but it's in fact it's two slightly different reading frames, and so there's a frame shift that happens between the two. And we'll take a look at that a little bit later on, how that happens. So what are some of the proteins there? There are a whole bunch of them. Don't expect you to remember all of them, but some of the important ones, again, have circles on them, and at least now it's the right table as opposed to the wrong one, as we had last time. So again, not terribly surprising, we've got viral proteases, NSP3 and NSP5. NSP5 is a lot like a picoRNA virus protease, and you can in fact see that here, 3C-like protease in number 5, and PLP1, PLP2, these papain-like proteases. Just like you have in the picoRNA viruses and the flaviviruses, these proteases have to be functional as part of a polyprotein. So again, as soon as the genome comes in, it gets translated. And that translation leads to the production of these proteases, which will chop things up. There's, and in fact this should be knocked out, predicted RNA-dependent RNA polymerase. This is the main RNA-dependent RNA polymerase. And probably the most important of these is NSP14. This is, again, non-structural protein, but this is the protein which seems to be actually giving the replicase some error-correcting activity. And so a lot of people predicted, seeing the size of the genome, that there has to be some kind of proofreading that's going on, some way of repairing incorrectly incorporated bases. And this is where this whole idea came up of, would you be interested in a vaccine that's got a much higher mutation rate? Because it turns out if you knock out this gene, the mutation rate goes up, and it goes up a lot. Still get functional virus, which is interesting, but mutation rate goes way, way up. And people are even trying to use this for a SARS vaccine, to take the SARS virus, knock out this NSP14, and then have that as a potential vaccine strain. So that's those are the names here. Let's take another quick look at them. Here, again, the 3C-like protease, NSP5. NSP3 is the main protease activity. The RNA polymerase, or replicase, here, gene 12, and then gene 14, which is this exonuclease activity. These guys are all in sort of the extended open reading frame. So how do you get that extended open reading frame? That's because, so let me move this here, pardon me. The RNA has a specific sequence that forms what's called a pseudoknot. And all that is a secondary structure that forms in your RNA, self-complementary, but basically instead of one stem loop, so you think about this top part as being a stem loop, part of that loop structure actually will bind to the stem of the next stem loop that's right next to each other. So basically you have two stem loops that are cross-linked through hydrogen bonds to each other. And this ends up looking like a pretty continuous double helix, and we'll look more at these structures a little bit later on. But these pseudoknot structures form right here in your genomic RNA, and of course the ribosome is coming along to this. And in 60% of the cases, the ribosome falls off. But in 40% of the cases, it keeps going and makes the rest of all of these genes here. So you don't need as much of your replicase proteins. You probably need more, certainly earlier on in the process, of your protease proteins in terms of making everything that you need to be able to replicate your genome. PLP1 and 2 are really the ones which are most important for early proteolysis, and NS5 is important for later proteolysis, particularly of these genes right here. So where do you replicate? Again, just like we've seen with the picoRNA viruses, with the flaviviruses, all these guys replicate in vesicles in the cytoplasm. I didn't upload the structures, but if you just look at a coronavirus-infected cell, it's filled with all kinds of really bizarre-looking vesicles. that might be associated with autophagy, which is sort of the cell eating itself. And it seems that some coronaviruses will actually induce this so that you have a lot more of those membrane compartments so that you can get replication happening right next to them. You have a lot less of the negative strand than you have of the positive strand. Again, this is not terribly surprising. Quite how that happens is not completely clear. Probably has to do with this propensity of the RNA-dependent RNA polymerase to jump around. And that has to do with recombination, which was getting back to thinking about these trees a little bit. So recombination, in terms of how replication is going on here, is basically the polymerase will be moving along one template and then switches over to a different template and continues. And what it's making, the anti-genome or copying into the genome, can then be a recombinant of the different pieces that you have in there. And again, we'll see this is what happens for retroviruses, things like HIV. So that's what's happening in terms of genome replication. Get a little bit of your negative strand and then a whole bunch of positive strand. And again, this kind of makes sense in terms of how we're doing replication and translation at the same time. Everything's going from 5 to 3, positive strands, translating your genome as well. But now you need to make capsids. How do you make capsids? Well, the way you make capsids is through all the structural proteins, which are down here at the 3' end of the genome. So this was a real puzzle for a long period of time. People eventually were able to purify the RNAs that are coding for each of these different structural genes over here, particularly this M gene, because that's the one that we've got the most of the protein for that makes the rest of the capsid structure. And what they found was, okay, all these guys had caps on them, which is not terribly surprising because you have a capping enzyme, which is part of the non-structural enzymes. But more importantly is they had this sequence at the end of each of these, which is identical to the sequence right next to the 5' cap structure. So the question came up is, how the heck is this happening? And so this gets back to recombination. What's happening here? So what seems to happen, and again, it's not completely proven, but it's pretty strong evidence right now, is that it turns out that there are these things called TRSs. The sequence here is not important. You don't need to remember it. But basically, these are sequences that when the RNA-dependent RNA polymerase gets to, it basically can either jump to the next one, or in many cases, jump all the way over to this one, or also jump from one strand to another strand in order to get recombination that's happening during your replication process. So as you're making negative strand, which is what's shown here as the brown, you get to one of these TRS sequences, again, starting from the 3' end, and you have a certain frequency of either jumping to a different strand, which you may have a different sequence because you still have a pretty high mutation rate, or jumping over to this TRS sequence at the end. This seems like a really long, this is 30,000 nucleotides. How are you going to be jumping from one end to the other? But you're never going to have a genome all stretched out like this inside the cell. They're all going to be associated with these vesicles. So the distance between the 3' end and the 5' end is probably not that far. Once you've then done this, starting, and all of them start from the 3' end, making your negative strands, you'll end up with a bunch of what are these called subgenomic negative strand templates. Now, each of these have the complement of the 5' end of the genome, because this polymerase gets to the end, will fall off at the end. This now can provide you sequences here, complement, to be able to make each of these subgenomic messenger RNAs, and these guys are what are being used to make protein. Now, how you go from making whole genome to making a whole bunch of these little pieces is still an open question. People are trying to figure out what's going on with the replicase in terms of these coronaviruses. But this is pretty clearly exactly how this is happening. Making subgenomic, so an RNA copy of part of your genome, and then copying each of those to make messenger RNAs. Why do you think the virus, again, to totally over-anthropomorphize, would want to have sequences at the very 5' end of your genome? What do you think those would be important for? Protection is one thing. What else? Packaging? Are you going to want to package these messenger RNAs? No. Why do you want to have this sequence at the 5' end? Okay, there's a cap there. That's actually the capping enzyme, which has put it on there. What else? What else do you need right there? So ribosome and translational regulatory sequences. So these guys don't have irises. They actually have cap-dependent translation. But there's also lots of sequences here which are really important to get translation to take place. So the translational regulatory sequences, which, not surprisingly, are really good, just like we have with all of the other RNA viruses. You get much more translation of your viral proteins than you do of cellular proteins. That seems to be what these particular sequences are, and evidently it's evolved, these particular viruses, in order to take advantage of that. How do we get genome packaging? Genome packaging here is actually extremely similar to what happens with the flaviviruses that we talked about last time, only instead of having an icosahedral structure, it's really much more of a helical nucleocapsid structure. So you've made your genome that gets coated with your nucleocapsid protein that interacts with your M protein, which grabs spikes, and you end up butting into the Golgi. So you've got a membranous enveloped virus inside the Golgi, and then that gets transported to the outer membrane and is basically released as you have vesicle fusion, as if you were beginning to be releasing some other kind of vesicle from going outside the cell. So these are fully formed virions, and they just show some cases here, icosahedral. Most of them are much more like this structure here. And you can, they say you can see donuts in the EM. I've yet to see an EM that actually shows me donut structures like this. But that's the process on how we get release. So that's the whole process, the whole replication process. There are a couple of things that recently people have found in order to make studying these ridiculously ginormous RNA viruses a lot easier. One of them is what's called reverse genetics. And the basic idea here is you need to make a RNA that has not just the normal RNA that you have from the virus genome, but so you can make mutations in it. So you can make mutations like this NS14 deletion that I was telling you about to see what happens when you get rid of that one particular gene. How do you go about doing that? Well, one of the easiest ways to do that is to take your original wild-type genome up here and make DNA copies of each of the different pieces of the genome. If you do this in a smart way, it's in lots of different pieces. You can modify one of those pieces, and that's what this X is shown here for. Then take all of these pieces of DNA, because DNA is a heck of a lot easier to work with than RNA, and fuse all of them together. The way you do that fusion is you just pick your favorite restriction endonuclease. Say this would be a restriction endonuclease EcoR1 at this end here, EcoR5 at that end, EcoR5 at this end, BAMH1 at this end, BAMH1 at that end. Chomp all of these DNAs out, ligate them together, and then put everyone's favorite promoter at the end here, bacteriophage T7, add bacteriophage T7 RNA polymerase, because it'll bind to the end, and makes this wonderful RNA, which has the change in it that you happen to make. And again, we'll just take the example of the deletion of NS14. Now, because these are positive strand RNA viruses, all you need to do is take this RNA, which you've made in vitro in the test tube, put it into a cell, and then look at the viruses that come out. Because all you need to do is get that RNA inside the cell. electroporation. Again, people who are in the recombinant DNA techniques lab, you'll be doing that this afternoon, but not with coronaviruses. Much more interesting viruses. These are, our sulfolobus viruses. So that was it for coronaviruses until last year. And that was the MERS. Actually, MERS was originally found in 2012, but got a lot more press more recently. So wanted to give you an update. 180 cases of MERS. Now, MERS is Middle East respiratory syndrome. At the beginning, people all thought it was SARS had come back because, as I mentioned before, last reports on SARS were 2004, 2005, 10 years ago. We're over that. Don't have to deal with it anymore. Well, 2012, we had the first reported case of the Middle East respiratory syndrome. Similar kinds of symptoms, again, pneumonia, hypoxia. 180 cases January of last year, went up to 1,000 as of this year. So many, many more cases. And we'll see why that is in just a second. Again, big outbreak in 2012, 3 of them in the US, about a 50% death rate. They isolated this virus at the Erasmus Medical Center. Anyone know why Erasmus Medical Center is infamous or famous? We'll talk about some flu experiments a little bit later on. Gain of function experiments, looking at transmission of flu between different organisms, in this case particularly ferrets. Wonderful place for doing a lot of this virus research. And they did the sequencing in 2012. Again, about 3 months between having a case and then isolating the virus and getting the genome sequence. 3 months, again, pretty amazing. Turns out it's closely related to some bat coronaviruses, but not to SARS. Really quite different in terms of the SARS sequence. And I don't have any phylogenetic trees. I could pull those up. And they found the receptor. Turns out to be another one of these proteases, extracellular proteases. And let's see, I think we've got a couple of minutes here. Let me see if this link actually is going to work. The last link that I had here didn't. But we'll take a look at this one. See if this will actually work. Don't know if we've got the sound here. The sound is just too good for this. SARS-like virus, death toll. Okay, let's see if we can actually get